All right, here we are at the second part of Chapter 10, and we will be discussing the cell cycle regulation and some checkpoints, talking about how mitosis gets co-opted and disrupted that can lead to cancer, and a brief description of binary fission or cell division in prokaryotes. Last time I misspoke, we are doing meiosis this week in lab. The weeks start to blend together after a while. And we will be talking about meiosis next week in lecture. We also have our second exam coming up next week, and the study guide for that will be posted today. And that next exam will cover chapters 7, 8, 9, and 10, and that means of recovering cellular respiration, photosynthesis, cell signaling, and mitosis and cell cycle regulation. This image is the cell cycle. We saw it earlier in the previous lecture, and now we've got not just the labeled interphase consisting of G1, S, and G2, and the M phase, or the actual cell division phase, but we also have these boxes labeled checkpoints, because the cell needs to have some way of detecting, is everything okay? Do we have enough nutrients? Do we have enough of the correct organelles? Is the DNA intact? Has the DNA been damaged in any way? All of that needs to be confirmed before the cell exits the G1 phase and enters into the DNA synthesis stage. So that's what's meant by the integrity of the DNA. Does the cell have all of the chromosomes it's supposed to have, or all the chromosomes intact? There's no duplications, there's no deletions, there's no damage. During the synthesis phase, that means the DNA was double-checked, everything seems to be in order, and DNA replication can occur. As mentioned last time, it's not just the DNA that replicates. The cell will also have to duplicate all of its organelles. But without the DNA, none of the rest of it can happen. Once synthesis is complete, we move into that second stage of growth, the G2 phase, because the cell does need to get a little larger before it divides. Otherwise, the cell size diminishes with each division. But before we can move from the G2 into the M phase, there is a second checkpoint that does assess that the chromosomes got duplicated correctly. Every single chromosome is duplicated. Every single duplicated chromosome has its sister chromatid attached at that centromere. And remember, 2 chromatids attached at the centromere is 1 chromosome. Flashcards. It's the only way to keep those straight. If the cell passes the test at all of its correctly formed DNA duplicated correctly, then it will start the mitosis phase. We'll go into prophase, where the chromosomes condense, and the mitotic spindles, made of microtubules, will attach at the kinetochores. And then we check at the M checkpoint during metaphase to make sure that the microtubules aren't only attached to each chromosome, but there's a microtubule from each side attached at the kinetochore, at the centromere, for each chromosome. If there's only a microtubule from one side or the other, then all bets are off, and the cell is not going to complete the M phase. Because if there's a kinetochore only at one side, then both chromatids are going to go in that direction. We need kinetochores on either side so that at anaphase, the chromatids pull apart and that we have equal distribution. A kinetochore attached only on one side to a microtubule means that both chromatids go to one side, which means they will separate at telophase, and now we'll have 2 copies of the same chromosome instead of 1 copy of each chromosome. One of the things that controls this are levels of proteins called cyclins. And they're called cyclins because they help regulate the cell cycle. We have A, B, D, and E. Not sure why there's no C. And they peak in their concentrations at different points in the cell cycle. So direct correlation between that cyclin accumulation and the cell cycle checkpoints. So cyclin E peaks at that G1 checkpoint. We have the most of that cell cycle regulator right here when we're deciding, does the cell have good DNA? Is this a cell with proper chromosome number and not damaged DNA? And then it passes that test and the levels of cyclin E immediately drop off as we're in the S phase. Cyclin A is peaking right to this G2 phase because this is where you have done the synthesis in the S phase. Now this cyclin has been building up and will stop things if the DNA did not replicate properly. If it did, then levels of cyclin A immediately drop, and cyclin B has been building up, and it peaks at that M checkpoint. We had the right DNA. Did we, in the process of prophase, correctly get a kinetochore attached on either side to its microtubules? Cyclin D starts out very low. It peaks in the S phase, and as the checkpoints are passed, its levels drop back off. And it's not just the cyclins by themselves that are carrying out these checkpoints, building up as the checkpoints are reached. We have chemicals called cyclin-dependent kinases. And we've mentioned kinases before in our figures for cellular respiration. A kinase is an enzyme that catalyzes the transfer of phosphate groups. So a cyclin-dependent kinase is an enzyme that's going to transfer phosphate groups, and that activity depends on binding to a cyclin. So when these cyclin-dependent kinases, or CDKs, are activated, they in turn phosphorylate other proteins, and that's what tells the cell to move past the checkpoint. So the cyclin-dependent kinase has to bind to a cyclin. The kinase itself gets phosphorylated, and then the CDK can transfer a phosphate to another protein. So here is some cyclin. Each cyclin has its own kinase that is dependent on it. So each cyclin has its own CDK, its own cyclin-dependent kinase. When that interaction happens, a separate kinase will act on the cyclin-dependent kinase and add a phosphate. That's what the little P with the circle is. Now, this complex is activated, and the CDK is an enzyme, and it will catalyze the transfer of the phosphate from itself onto a target protein, and that target protein is the cell signal that says we can move past this checkpoint in the cell cycle. Each of these cyclins has its own cyclin-dependent kinase. When the cyclin binds its cyclin-dependent kinase, a separate enzyme puts a phosphate on the cyclin-dependent kinase, and then the CDK can catalyze the transfer of the phosphate on itself to the target protein, and that's what signals move past this checkpoint in the cell cycle. The RB protein is another key regulator in the cell cycle. It was the first tumor suppressor gene that was discovered. A tumor suppressor gene does what it sounds like. The goal of that gene, the product of that gene, is to suppress the formation of tumors. Tumor is unregulated cell growth. We can have benign tumors where there's just reproduction of cells and there's just extra cells that forms a lump. Could be anywhere in the body. Skin tags are benign tumors. Moles are benign tumors. You could also have things like a lump in a breast. Might be completely benign and have no cancer cells. But they could also be duplications of damaged cells, in which case that does become malignant or cancerous. It is called retinoblastoma, the RB protein, because it was first discovered in patients that had retinoblastoma, which is a tumor of the retina. So blastoma or oma will always tell you that there's a tumor, and it's a retinoblastoma. It happens in the retina. This is genetically inherited, and only one copy of the damaged gene for the RB protein is necessary for the person to suffer from the cancer. The way it works when you have a normal RB protein is that it is bound to a transcription factor called E2F, which is elongation factor 2. This factor is what will bind to DNA and allow for gene expression to happen. So if retinoblastoma protein is bound to the signal for the gene to be transcribed or to be turned on, then there is no transcription, there is no cell division. But if there are signals that this cell does need to divide, then we hydrolyze 2 ATP molecules and put 2 phosphates onto the retinoblastoma protein. The binding of the 2 phosphates causes a conformational change, and the E2F is released from RB. The E2F binds to the DNA, and that turns on the gene expression that allows the cell cycle to continue. If the retinoblastoma protein is damaged, one of 2 things could happen. It's either unable ever to bind the E2F, and so we're always getting the signal to move forward with cell division, or once it gets phosphorylated, these phosphates never get removed, and we keep it in this form that releases the E2F and says, keep growing. The other really important cell cycle regulator that we will discuss, there are more than just these 2, but these 2 are the best known. The second one is called P53, because it's the 53rd protein that they were discovering. The normal P53, if there is DNA damage, any sort of cell cycle abnormalities, we're continuing to move through the process, we're unable to move through the process, the cell cycle just isn't working the way it should, or in conditions of hypoxia. Hypo means not enough, and oxia is oxygen. The cells are starved for oxygen. When cells are starved for oxygen, they will do crazy things to try to keep surviving, and we don't want damaged cells to continue. So any of those conditions in the presence of a normal P53 will stop that cell cycle. If possible, do DNA repair. All cells have enzymes that can proofread, go back and look at the DNA sequence, make sure that any damage has been repaired. And if repair can happen, then the cell cycle starts again. If we cannot repair the DNA, we will not continue the cell cycle, in which case we arrest the cell cycle, it stops, and then we get apoptosis, programmed cell death. It is a naturally occurring function in our cells that if it is damaged and non-functional or could potentially lead to unregulated cell growth, P53 is patrolling everywhere in our bodies, excuse me, to get rid of those cells, to program them to die so they cannot multiply and cause problems. However, if the P53 protein is damaged, then despite the signal that there is something wrong with the DNA or the cell cycle or there's not enough oxygen, the cell cycle just continues, and if we have damaged cells, that's where we can get cancer because we have unregulated proliferation, cell division going on, and it's not regulated. And our final slide is just looking at binary fission. Prokaryotes are single cells. This one is showing a cell that has both a plasma membrane, because all cells have a plasma membrane, and a cell wall, because most prokaryotes do have a cell wall. There isn't a cell cycle, really. We don't have mitosis in prokaryotes because there's only one chromosome and it's a circle, and they don't have microtubules for pushing and pulling their chromosomes. So there is an origin of replication, the start of where DNA replication happens, and the FTSZ proteins will migrate toward the midpoint of the cell, and they are what will help to cause the cleavage furrow and split the cell in two, and then we can form the cell wall. In animal cells, we have actin filaments that will split and cause that cleavage furrow. In plant cells, we have Golgi vesicles containing cellulose that will split the cell so that each cell has a plasma membrane and a cellulose cell wall. And in prokaryotes, we have FTSZ proteins that migrate and pull the edges in together so that each new cell has its cell wall and its plasma membrane. That's a super brief lecture because we've got some pretty intense chapters. Didn't want to overload this week. This lecture material will be the end of the material for exam 2. Again, that will cover chapters 7, 8, 9, and 10. The study guide will be posted by tomorrow, which is Wednesday, because I'm recording this on Tuesday. And it will be similar to the layout of the first study guide. The exam itself will also be the same format as the first exam. So for better or for worse, at least you know what to expect. There will be definitions, there will be multiple choice, true, false matching, there will be short answer, and then a choice of thought questions. So you have a week to work on those thought questions to develop a fully researched and supported answer to the question. That's worth 30 points. And that means you have a week to discuss with each other, to discuss with me, to have me read it over and make sure you've gone into as much depth as you need to. So I will send out an announcement when that study guide is ready, and it will also be posted in the Module 4 lecture exam study guides.